CRISPR breakthrough, one amino acid edit blocks malaria in mosquitoes. Imagine a world where a tiny genetic tweak could turn the tide against one of humanity's oldest and deadliest foes, malaria. Sounds revolutionary, right? But here's where it gets controversial. As mosquitoes develop resistance to insecticides and malaria parasites outsmart our best drugs, our public health arsenal is struggling. However, a groundbreaking study published in Nature offers a glimmer of hope. Researchers have unveiled a novel genetic approach to halt malaria transmission, modifying a single amino acid in mosquitoes, rendering them incapable of spreading the parasite. This strategy includes a self-sustaining allelic-drive that spreads this resistance throughout wild mosquito populations. The study, titled Driving a Protective Allele of the Mosquito FREP1 Gene to Combat Malaria, https://dx.doi.org/1038/S4-1586-025-09283-6, presents a third pathway in genetic malaria control. Unlike methods that eliminate mosquitoes or introduce foreign genes, this approach reshapes mosquito populations from within. Ethan Beer, Ph.D., a professor at the University of California, San Diego, explains, "This is a third genetic strategy to combat malaria. The existing methods involve either reducing mosquito numbers or modifying them to produce transmission-blocking antibodies. This new approach, however, restructures the existing population, replacing efficient malaria transmitters with inherently resistant mosquitoes. The focus is on the mosquito gene FREP1, crucial for blood feeding and a key entry point for Plasmodium parasites. https://www.geningnews.com/?S=Plasmodium into the mosquito's salivary glands. By changing one amino acid in FREP1 from leucine, L224, to glutamine, Q224, the researchers created a variant that stops parasite transmission without harming the mosquito. The idea that altering a single amino acid could make mosquitoes resistant to malaria parasites was a startling discovery," Beer told Jen. Another surprise was the efficiency of their drive system, which exceeded expectations due to a mechanism that eliminates individuals carrying the gene cassette but not the preferred FREP1Q allele as a byproduct of CRISPR activity. Developed by Beer's lab in collaboration with George Demopoulos, Ph.D., at Hopkins University, the allelic-drive system uses a CRISPR-Cas9-based gene editing cassette. Unlike standard gene drives, its components can be placed elsewhere in the genome, offering greater flexibility and the ability to self-eliminate once the task is complete. Beer highlighted this crucial difference. The gene cassette encoding the CRISPR components can be placed at a different genetic site than the one to be acted on, while the gene drive must copy itself at the location it is inserted into the genome. For instance, the system can be engineered to disappear, leaving a population inherently resistant to malaria infection. In contrast, a gene drive expressing transmission-blocking antibodies would need to remain in the population to be effective. The team directly linked the guide RNA, gRNA, targeting the non-preferred FREP1, L224 allele to the preferred Q224 variant, ensuring the gRNA is never inherited without the desired protective allele, thus avoiding unwanted mutations, Beer explained. The most innovative aspect was this linkage, Beer noted. If such a separation occurred, the drive system could copy an undesired mutant version of the gene, reducing its effectiveness. Our system prevented this, and no unwanted mutant alleles accumulated during our experiments. Tests on Anopheles stephensi, a significant malaria vector in Asia, demonstrated that the single amino acid change blocked 2 parasite species from reaching the mosquito's salivary glands. Importantly, the edited mosquitoes reproduced and developed normally. The beauty of this approach is leveraging a naturally occurring mosquito gene allele, said Demopoulos. With a single, precise tweak, we've transformed it into a robust shield against multiple malaria parasite species. While the FREP1Q linked allelic drive isn't suitable for Aedes mosquitoes, which spread dengue, the platform is highly adaptable. The system is broadly generalizable to any situation where efficient gene drives can be generated, Beer stated. The only requirement is the presence of two functionally equivalent gene variants. Looking ahead, Beer envisions combining this strategy with traditional public health measures for maximum impact. We don't see any genetic system as a silver bullet, he said. Integrating suppression drives, modification drives, and allelic drives with tools like bed nets, insecticides, and antiparasitic drugs could create a far more potent system. An appealing combination would be to first suppress mosquito populations and then modify the remaining few to prevent parasite transmission. This project exemplifies the power of interdisciplinary, international collaboration. Beer's team worked closely with Dimopoulos Lab, FREP1 biology experts at Johns Hopkins, and mathematical modelers from the University of California, Berkeley, and the University of Sao Paulo. This breakthrough is the result of seamless teamwork and innovation across institutions, said Dimopoulos. Together, we've harnessed nature's own genetic tools to turn mosquitoes into allies against malaria. So what do you think? Is this genetic tweak a game changer, or does it raise ethical concerns? Share your thoughts in the comments. Gene editing breakthrough. Can we stop mosquitoes from spreading malaria? Imagine a world where a tiny genetic tweak could save millions of lives by stopping mosquitoes from spreading malaria. Sounds like science fiction, right? But here's where it gets groundbreaking. Researchers have discovered a way to modify a mosquito's DNA so subtly, changing just a single amino acid, that it could dramatically disrupt the malaria parasite's journey to its next victim. And this is the part most people miss. This small change could be the key to transforming entire mosquito populations into ones that can no longer transmit the disease. Every year, malaria infects 263 million people and claims over 600,000 lives. The parasite's path to infection is complex. It must first enter a mosquito's gut when the insect feeds on an infected person's blood, then travel to the mosquito's salivary glands, ready to be injected into the next victim. But a team of scientists has found a way to interrupt this critical process. By using CRISPR gene editing technology, they've altered a gene called FREP1, which plays a role in both blood feeding and the parasite's journey. The result? Mosquitoes with this tweak carry significantly fewer parasites, up to 90% less, making them far less likely to spread malaria. But here's where it gets controversial. The researchers plan to use a gene drive to spread this modification through wild mosquito populations. Gene drives are powerful tools that ensure a specific genetic change is inherited by nearly all offspring, rather than the usual 50%. While this could revolutionize malaria control, critics like Dana Pearls of Friends of the Earth warn of unpredictable consequences. Gene drives could permanently alter ecosystems, she says, raising concerns about unintended mutations or ecological disruptions. For instance, what if these changes affect other species like birds or bats that rely on mosquitoes as a food source? Proponents argue that new genetic tools are necessary because traditional methods like bed nets and antimalarial drugs haven't eradicated the disease. George Dimopoulos, a biologist and study co-author, emphasizes that this approach isn't a quick fix but a long-term strategy. These mosquitoes will spread naturally, gradually replacing malaria-transmitting populations with ones that can't spread the disease, he explains. Yet, it's far from ready for real-world use. Field tests are years away and require community and government approval. What makes this approach unique is its subtlety. Unlike other strategies that introduce entirely new genes or aim to wipe out mosquito populations, this one tweaks an existing gene in a way that's naturally occurring in some mosquitoes. It's marginally more acceptable to those wary of genetic engineering, notes Anthony James, a molecular biologist not involved in the study. But the question remains, are we ready to rewrite the genetic code of an entire species to fight a disease? And what if the malaria parasite evolves resistance to this new tool? This research opens up exciting possibilities but also sparks critical debates. Should we manipulate nature to save lives, even if the outcomes aren't fully predictable? What safeguards are needed to ensure these tools don't cause more harm than good? As scientists push the boundaries of genetic engineering, these questions become increasingly urgent. What do you think? Is this a bold step forward or a risky gamble? Let's discuss in the comments.